This is Maya. Growing up, Maya started noticing that some of her features were not like her parents. Both her mother and father had brown eyes, while hers were blue. Her hair is also light blonde, while her parents have dark brown hair. Eventually, Maya's friends started to notice, and they began making fun of her for the striking differences in characteristics between Maya and her parents. They would laugh at Maya, saying she must be adopted or even switched at birth. This made Maya really sad, and actually start to question herself as well. To combat her friends' comments and her own feeling on the matter, Maya searched for a solution to find out once and for all whether she was actually adopted or not. Maya went to her science teacher for some assistance with her genetic predicament. And, with the help of her science teacher, Dr. Wang, Maya was able to finally discover the key to solving her problem. Maya had found that the solution lies within her DNA. Maya found that DNA, short for deoxyribonucleic acid, is the molecule that encodes critical biological information in humans and other organisms. And, since half of the DNA from each parent goes to their child, Maya discovered that decoding her DNA would prove her hypothesis. With this new information, Maya began searching for a way to decode her DNA that was quick, easy, and affordable through some online research. This is where she found Nanopore Sequencing by Oxford Nanopore. Nanopore Sequencing is an affordable, real-time DNA/RNA sequencing biochemical sensor. The device can sequence any length of DNA/RNA strand, is scalable to the user's needs, and provides rapid results. Maya found this to be a perfect solution and sought to figure out how it worked. Nanopore sequencing begins with the nanopores themselves. Nanopores are tiny holes within the biosensor that DNA/RNA molecules flow through. The nanopores are embedded in an electroresistant membrane, and each nanopore has its own electrode and sensor chip. The structure of the nanopore begins with a motor protein that pumps the DNA/RNA strand through to the actual nanopore. This is known as the nanopore reader. The DNA/RNA then hit the electroresistant membrane. As the DNA/RNA molecules flow through the nanopores, the specific ionic currents associated with each strand of DNA/RNA is recorded through an RNN algorithm software. This reading gives what is called a squiggle, of which is the raw data associated with each strand. The raw data readings are then written out by and stored as POD5 files. These files are then decoded using a RNN algorithm to convert the POD5 files to FESTQ/BM files. Finally, the files are processed through an integrated base caller called Dorado. This process is known as base calling, where the electrical signals generated by a DNA/RNA are processed into their corresponding base sequence. Each strand of DNA/RNA has a specific set of bases associated with it in a sequence of A, G, T, and C, which differentiates each strand from the next. The base calling algorithm is like a brain. The nanopore sequencing is modeled by biological neural networks in the brain, where nodes equivalent to the brain's neurons pass data between themselves. A bidirectional recurrent neural network is used, where information can go back and forth between nodes. The data from the base calling is presented to the user, where they can analyze the results for different uses, such as forensic science, ancestry, disease surveillance, and biological research. Or, however, in Maya's case, though nanopore sequencing could help her in numerous other fields, she just wants to learn if she could be adopted or not. And so, through the process of nanopore sequencing, Maya was able to determine that she was not adopted. Instead, Maya was just a very unlikely genetic result of her two parents that happened to have very different traits than her. And so, at the end of the day, finally, loaded with this new information, Maya was able to prove to her quote-unquote friends, once and for all, in confidence that no, she was indeed not adopted. And this was all through the power of nanopore sequencing by Oxford Nanopore. Thank you.